Hello, my name is Russ Sashtaki, and I'm a biological sciences tutor here at Winston-Salem State University, and today we're going to go over the cell. So the example that I have here is a cell from an animal. So that could be a person, that could be a dog, that could be a cat. It's considered a eukaryotic cell, which means it has a nucleus. If a cell does not have a nucleus, it's considered to be a prokaryotic cell. Like bacteria, for example, that's a cell that does not have a nucleus. It's considered a prokaryote. But this is an example of a eukaryote or eukaryotic cell. So let's get right into it. All right, so when you're looking at this cell, this is the nucleus. The center of the nucleus of the cell contains DNA. Now, the DNA is your genetic material. It has all the information that you need to code for you. So all of your cells have all of your DNA, and you need to protect that. So the nucleus has a nuclear envelope, as you can see here, that protects the DNA. Now, when you need to copy some DNA to create a protein or make something, you will take that DNA, copy it into RNA, and that process is called transcription. Then you will take that RNA out of the nucleus through these little tiny nuclear pores. There's little tiny holes that allow for the RNA to leave. That RNA then goes to a ribosome, which is usually on the rough endoplasmic reticulum. Ribosomes make the rough endoplasmic reticulum rough, and that is found outside the nucleus here. You will see on the ribosomes, all of those have little tiny dots on the rough ER, and those are the ribosomes that make the proteins. The process of taking the RNA out of the nucleus to the ribosome to make a protein is called translation. So you need to know the difference between transcription and translation. The other thing that's really important is the center of the nucleus, which is here. The center of the nucleus in the middle is called the nucleolus or the nucleolus, depending on where you're from. I call it a nucleolus. The nucleolus is the center of the nucleus, and its primary goal is to make the ribosomes. Ribosomes come in two units, large and small subunits. Just like when you get furniture, they don't deliver it to you in one piece. Usually it comes in a box, especially if you get it from IKEA. So you get that furniture and then you assemble it in your house. Because if it's something large, like a dresser or a giant bookcase or a table, you probably won't be able to get it through the front door. So you bring it in pieces. Same thing here. The center of the nucleus, the nucleolus, has the ability to make ribosomes, but it makes them in pieces. It has a large and small subunit that it makes. Those two units can then leave the little pores outside of the nucleus to allow for them to assemble themselves out on the rough ER or in the cytoplasm, which is a liquid part of the cell. So if you try to get that assembled ribosome out of those nuclear pores, it's too big and won't go through these little tiny holes on the nuclear membrane. So then you put them in large and small subunits, and then you take them out. Again, ribosomes can be free-floating, or they can be in the endoplasmic reticulum on the rough endoplasmic reticulum, or the rough ER. Ribosomes make proteins. Remember that, okay? My name is Russ. You can remember that Russ says ribosomes make proteins, and they are found on the rough ER. Ribosomes make the rough ER rough. So hopefully you won't forget that. And then the smooth ER, the smooth endoplasmic reticulum, is devoid of ribosomes. It has none. So therefore it makes everything else but the proteins. So carbs, lipids, things like that are made in the smooth ER. Okay? And then this is the Golgi. The Golgi apparatus packages things. The Golgi's main goal is to package things. It's like the UPS center or the mailbox, etc. store or the post office, okay? FedEx office, any of those places. So what happens here is you package things and you package them in that Golgi. And once you do that, then you send them out either for excretion through what they call exocytosis, or you can use it within the cell, okay? Remember, when you're bringing things into the cell, it's called endocytosis. When you're sending stuff out of the cell, it's called exocytosis. So the Golgi packages things from the rough ER, like the proteins, or the smooth ER, the carbs and lipids, and parts of the cell membrane are made. And that goes into little vacuoles, vesicles, that basically carry them out through exocytosis, okay? So the Golgi packages things. And then the next major organelle is this, okay? These little kidney bean looking things called the mitochondria, okay? There's one there, there's one here, there's one there, there's several of them. Especially nerve cells and muscle cells, they make the most amount of energy because they require the most amount of energy. The reason why you eat, the reason why you breathe is to make ATP, which is energy in the mitochondria. You consume food primarily, your mitochondria love to run on glucose, and the oxygen that you breathe goes into these mitochondria, and out comes ATP, CO2, which you breathe out, and water vapor. So if you ever, exhale on a mirror, for example, you would see it fog up. That's because you're exhaling water vapor and CO2. Those are the byproducts of what they call cellular metabolism, which we'll talk about in another video. So this basically is your energy source for the cell, all right? And at one time they say that our cells engulf in mitochondria, and that's why mitochondria have double membranes. And mitochondria have their own DNA, so they used to live outside of us before. So animal cells took them in so that we can make energy for ourselves. These mitochondria primarily like to run on glucose. One molecule of glucose will give you up to 38 ATPs. That's a lot of energy. So especially for nerve cells, muscle cells, they're very, very important. So when you think of mitochondria, think of the powerhouse of the cell making energy. And like I said, when we talk about cellular respiration, we'll talk about what goes on inside that mitochondria, okay? Another thing I want to talk about are these little centrioles. These centrioles here, these little star-like looking things, they are used in cell division. Their primary goal is to go to the opposite ends of the cell during what they call anaphase, which is the phase of mitosis, or meiosis if you're talking about egg and sperm. And mitosis and meiosis are cellular division, okay? And we'll talk about that some more in another video as well. Those centrioles then go to opposite ends of the cell during anaphase, and they pull the DNA apart to opposite ends of the cell. So when you copy all your DNA, you can separate them to either side. So then you can cleave it through cytokinesis, which is cellular cleavage. And then you end up with two new cells. You don't want to cut any of your DNA during the cytokinesis process. So metaphase, all the DNA lines up. Anaphase, that phase of mitosis is where the centrioles shine. That's where they do their work. They pull the DNA to opposite ends of the cell. And then once the DNA is separate, then you start the telophase and the cleavage furrow, and then the cytokinesis, which is complete cleavage of the cell. Then you got two brand new cells with two identical copies of DNA, and the nucleus reforms, and the cell goes about living its life until the next time it's time to divide. So these centrioles here, their primary goal is to separate DNA, and they're used during cell division, okay? And then the cell membrane, of course, is called the phospholipid bilayer, okay? Plants have a cell wall, animals have a cell membrane. The cell membrane has cholesterol in it. Plant cells don't have that. They're very rigid, okay? Phospholipid bilayer has hydrophobic, which is water-fearing tails in the center of the cell membrane, and hydrophilic, which is water-loving heads on the outside and at the out of part of the cell and the inner part of the cell. So wherever you have water, you will see the hydrophilic heads, okay? And again, we'll see in another video the cell membrane in greater detail. And then these little tiny peroxisomes and lysosomes, okay? So the peroxisome is there, the lysosome is here. The peroxisomes and lysosomes contain enzymes, and those enzymes break down things. Lysosomes break down things like food or bacteria or other organelles. So if you have a germ you want to destroy, or if you have food that you want to break down, kind of like in your stomach, you have stomach acid, lysosomes will engulf a piece of food through phagocytosis, which is large particle endocytosis, bringing something into the cell. And then once you bring that in, then you ultimately will break it down. And peroxisomes are the same thing. So peroxisomes also break things down using hydrogen peroxide, okay? So they normally are there for breaking down things and generate hydrogen peroxide. But anyway, so that's basically the cell in a nutshell. If you have any questions, please feel free to see me. I'm in Hill Hall on the first floor in the tutoring center, and I look forward to seeing you. Thank you.